present, please let us know so that we know how much time we are saving. Just to inform you that for those of you that would like to have the abstract booklet in electronic format, there is a barcode, the same one that I am holding. You'll find it outside there. And if you scan it, if you scan this barcode, you get the abstract booklet. So it is available. Please scan it wherever you have time. Again, to remind you, yesterday we indicated that we are looking for nominees for members of the executive committee of the ISCTRC. We have nominees from Eastern Africa, Western Africa, and yesterday we got a nominee from Southern Africa. So we are waiting for the nominees for Central African countries and for Northern Africa. We have only today. Please make sure that you meet as delegates from those countries, and by consensus, let us have your nominee, because these names will be presented to the delegates tomorrow for them to endorse those nominees to become members of the executive committee of the ISCTRC. I now would like to pass it on to the moderator, Dr. Dan Masiga, and the rapporteur, Dr. Kama Dramane, to take us through the rest of this session. Dr. Masiga.
Thank you very much, Chair. And thank you all for coming early. We want to just start from where we left yesterday. So we will be completing 5B, what is indicated as 5B in your programs, and then move on to 5C. 5C is on diagnosis. So if you are giving a talk during that session, please make sure that your slides are loaded. I'd like now to invite Nancy Ngari. Nancy is going to speak to us about efforts to develop the SSI Atlas in Kenya.
Good morning, ladies and gentlemen. Good morning once again. My name is Nancy Ngari from the Kenya SSI and Trypanosomiasis Eradication Council. And I'm still waiting for the IT team to give us a presentation. Yes. No, you have not started counting. Okay. Once again, I'm Nancy Ngare. I'll be presenting on the mapping the pathway for the progressive control of animal trypanosomiasis in Kenya. Okay. Yeah, so that will be the outline. Next. So as we all know, for the last many decades, a lot of work has been done in Africa on SESE and trypanosomiasis control. We have gotten a lot of data, we have gotten a lot of information, either lying in our offices or wherever it may be. But the big question is, how do we use that data for our good, to inform policy, for resource mobilization, to our politician for advocacy, so that it can work for us. All this research work that we are doing. Please, the previous slide. And the biggest question is, could PCP be a way that we can package this information for our advocacy and our resource mobilization? Next. So PCP are stepwise approaches, as FAO had explained earlier, for the control, for the elimination, and for the eradication of both livestock and human diseases. They are stepwise, as you can see, there are 5 stages, so you move from one stage to the other. But also the reverse is true, whereby you don't do what you are required to progress to the next stage. You are likely to go backwards. Next. For the PCP, there are 2 main, for the PCP, there are two main aspects. That is we have the roadmap. The roadmap simply shows you the big picture. That in a place X, for the next 10 years or a number of years, this is where I want to be. I'm here today, we are here in this stage today, but in the next couple of years, this is a picture that we are creating, we want to be in this given stage. So that is the roadmap. And then we have what we call the work plan. A work plan is the planning of a short duration of what you need to do for you to attain the big picture. So there is a roadmap, the first illustration there, and we have the work plan. So in this work, what we were generally looking for was mapping a pathway for the progressive control of animal trypanosomosis in Kenya, with the specific objectives being updating the databases of epidemiology and control activities on TNT, developing PCP maps from the databases, and developing a spatial explicit PCP work plan and roadmaps for Kenya. So, as earlier said by FAO, data is key in PCP mapping. So we have different types of data that we have worked around in order to come up to do our PCP mapping. One type of data is the epidemiology core data. That is basically the AAT and the SESE data, of which we have covered a period of 2000 to 2022. And this data is not only from Kentech or Patech, but also from stakeholders. Then we have data on control activities, the various strategies that Kentech is using in controlling TNT in Kenya. We have data on sustainable land management. These are areas that have been fed of SESE, and we have introduced sustainable land management, and this basically is through the promotion of artificial insemination. Then we have capacity building data, of which what we have mostly used is the stakeholder sensitization data. Then, with all these kinds of data, how do you work through, or how do you handle all this kind of data when you're doing your mapping? So a few aspects are very important when you're doing the mapping, that we consider the start date. We want to know, when did you start your programs? When did you do your baselines? When did you do your surveillances? Start date and end date. And of course, that gives us the period or the duration of which you have been working on that particular aspect. We are looking at the prevalence levels. What were the levels of the disease during baseline? What are the levels of the disease currently? What are the FTD levels during baseline, and of course at the current level? And when you're talking about current, it might not be a specific year. Mostly we were working with an average of around 3 years, because we know mostly we don't do our work year to year in the same place. So we're also looking at numbers. And with numbers, we are asking ourselves, for example, if you come and you do surveillance, AAT surveillance, and you're screening 100 animals, are they sufficient to cover a whole village livestock population? Then, so when we start working with all this data, we have to subject them to a criteria, so that to understand where the different places or the different areas are in the PCP staging. So we have a criteria for PCP, and it has stages 1 up to 5. But as we were working on our data in Kenya, we found that especially for stage 1 and 2, we had to subdivide it into 2. An early stage and an advanced stage. So for stage 1, what we are basically looking at is knowing the status of TNT in a given area. So 1 early is where you have not done any baseline, you have not done anything. So you don't know what the status are in that particular place. In stage 1 advanced, you have already done your baseline, and so you understand the status of TNT in that particular area. For stage 2, basically is you have introduced some control measures, and you have sustainably reduced the disease, the AAT disease prevalence. So stage 2 early, you have initiated your programs, but these programs have not yet bore any fruit. So there's no reduction in the disease yet. Then when you go to the stage 2 advanced, the programs have already taken effect, they have an impact, and you can see that the disease has started reducing, or the disease has reduced. And for the PCP, actually, we're looking at least 75% reduction. That is what we were calling considerably reduced. Then for stage 3 is whereby, after you have reduced the disease considerably, now you can introduce an elimination strategy. Then for stage 4, you have achieved your elimination, but you have your controls still in place. The control measures, the targets, the spraying is still going on. Then for stage 5 is whereby, yes, you have sustained that elimination, and at that particular time, there is no control of the disease that is going on. So having done all that, of course, we created our databases for epidemiological, that is for SESE and for AAT, and these are the maps that we got for the 2. And out of that, we published actually a paper earlier on, which was only based on Kentech-Patek data. Then we went ahead, also created databases for control, the various control programs that we have in the field. And also we have created maps for the various control programs that we have in the different areas that we are working in Kenya. Then from all these databases, we were able to create a PCP map for the country. And for this, we did for all the 47 counties, that is including the counties where there is no SESE in Kenya. And for this map, you can see like 66% of the counties, that is 31 counties, were in stage 1, either advanced or early. And then 16 counties, that is 34%, they were either in stage 2 early or 2 advanced. And for this PCP staging, as you can see, we have done for all the 47 counties. Then we went ahead and asked ourselves, because as we all know, county is the second level of administration in Kenya. That is like a district to many other countries, But we went ahead and we zoomed in. When we look at these counties, we all know that these counties, it's not the whole county that is SESA infested or has AAT risk. So we zoomed in to look at a lower administrative unit for some of the counties. And as you can see, there was a lot of interesting things, interesting scenarios inside those counties. Because, for example, on my far right, yeah, this one it's not working. On my far right, you can see a county that has 4 sub-counties, but it has 3 stages in it. So it's very interesting, and it shows us the variances in our counties, and the need for us to go even lower, at lower levels of administrative units or levels of working, for us to be able to get the picture, the real picture of what is happening in the areas that we are working in. Just hang. Then, apart from just the PCP mapping, we went ahead and created a PCP Smart Work Plan. This is a work plan for 1 year that we're asking ourselves for example, last year, that was the year 2022, a certain county is in a given level. How do we want to see that county in 1 year's time? So we created a work plan that is PCP Smart, as you can see on this, that map that is showed there. But as we are doing all this work, there are a lot of challenges in the PCP mapping that we encountered. One is about threshold. FAO said that the guidelines for PCP mapping are not yet out. So we having a lot of what is the threshold representation that is required in every of the entry that we are doing. What about variability in seasons and working in different geographic areas but within the same administrative unit? How do you counter for that? There is the issue of the conservation areas versus the surrounding areas. How do we map that? There's the issue of climate change. We all know some areas that were not SESA infested, right now they are SESA infested, where the risk of AAT was low, right? Some areas, they have a very high risk right now. How do we handle all that? And then at the end of the day, what is the way forward, or what was our conclusion on this? Our conclusion was on this. This is a strategy for Kentech. Not for Kentech, but for SESE for the next 10 years for Kenya. It's summarized in this table. For the next 10 years, where do we want to see each of our counties? So this is a big picture that we have printed for Kenya for the next 10 years. It's on a draft stage, so we are still working with it, on it with the stakeholders. And as you can see, some of the counties, we want them to progress very well to the very end. But others, we just want to maintain the status quo. There are some of the counties that we will not be doing actually anything on it because the risk of AAT is very low. So we'd rather concentrate on the other counties where there is much more risk. And for us, I think, for me, what I can say on conclusion is this is the way to go, because this is an evidence-based planning tool. This is an evidence-based resource mobilization that you can be able to show to people, to policymakers, to donors, that this is where we were a few years ago, this is where we are today, and this is where we want to be in the next couple of years. That said and done, thank you so much. I really want to thank our partners that we have worked with, both international and internal partners, and in particular the combat project as funded by the EU. Thank you so much.
Thank you, Nancy, for that presentation. Next presenter is online, Jean-Yves Ekra, who's talking about molecular epidemiology of trypanosomiasis in northern Cote d'Ivoire. So if you can please share your slides. Jean-Yves Ekra. Jean-Yves. And please go ahead. 10 minutes.
Hello.
Recording in progress.
Can I hear?
Yes, I can hear you.
Okay, all right, thank you. So, thank you everyone. I am going to present my topic. Sorry, this slide. No. Okay, I'm going to present my topic about molecular epidemiological survey of pathogenic trypanosome in natural infected people in the north of Cote d'Ivoire. So, I am going to follow this plan as introduction, bacterial method, result and discussion, and to end with a conclusion. In introduction, you can say that trypanosomiasis is one of the major complaints of livestock development in sub-Saharan Africa. Organisms of genus Trypanosoma transmitted mainly by the tsetse fly, which is a causative agent of the disease. So, trypanosome infection has a significant socio-economic impact and limit animal productivity in all African countries, including Cote d'Ivoire. The epidemiology of this infection has not been well covered in the League of Human African Trypanosomiasis, whose epidemiology is more mastered with some good research. Actually, in Cote d'Ivoire, we have been limited human African trypanosomiasis as a public health problem, according to Kone and Kaba 2021. So, however, epidemiology of animal trypanosomiasis is not well known in Cote d'Ivoire. So, reproductivity loss due to the animal African trypanosomiasis is around 30%. So, understand the status of this infection is essential to provide the knowledge necessary to appropriate control of African trypanosomiasis. So the objective of this work was to study the epidemiology of bovine trypanosomiasis, understand the distribution of trypanosome species, and also to know the most infected bovine in livestock. As a material and method, we are going first to present our study areas, which is shown by this map. This map is indicating is part of the north of Cote d'Ivoire, which is composed by three regions as Bagwe region, Oro region, and Chologo region. This study is conducted in a period from August to November 2021. And we can say that this area is also the most important part of Cote d'Ivoire where we have cattle farmers. To access this epidemiology, we did a cross-sectional research in cattle. Animals were randomly selected from each location. The total number of cattle in study was determined based on the previous studies that they already did about the prevalence. So as a data collection, before each collection, the animal were examined by visualization of vital sign. So we keep our goal on the animal which they are a poor vital sign, And the first sample was taken from the jugular vein with ISCTRC tube, and these tubes were kept on the cold storage until the molecular analysis was performed in the lab. The second sampling was done with a capillary tube from the hair of disinfection with alcohol. One was used for blood smear, and the second one was used for the analysis of hematological. In addition, the blood sample we take some information on the farmer as the breeder, the sex, and the position, the local of this farmer. So, the presence of trypanosome was determined based on the morphology using microscopy. Also, to assess the prevalence, we did a PCR, which is before that we did DNA extraction from the blood sample according to the commercial available kits. To assess that, we did the 3 different PCR. The first one is target the ITS1 region, which is a non-coding DNA between 18S and 528S. And also we perform a nested PCR, target the partial 18S, ITS1S, 5.8S, and ITS2, and partial 28S RNA. So to be sure for every trypanosoma that we get, we perform also some specific PCR with a microsatellite primer. So this formula is used for the prevalence, and to test the prevalence, we did a K2 test to know the relationship between our result and those factors. So, to assess the infection probability of an animal, we perform also a logistic regression with all of the factors as a parameter of our model. So this factor we can say that this factor, we have a packed cell volume, the race of animal, the region, the department, the age of animal, the sex, and also to end with the residual that follow up the normal distribution. So depending on all of all of these factors, depending on all of these several models were tested. The best model was selected according to the archaic information criteria. So for the for the molecular method, we use this logistic regression, and for the microscopy, we use this regression. Also, the meaning of hematocrit level were prepared by abnormal tests according to the infestation with a different consumer species. So all of this data analysis was performed with L, and also we use QGIS for the map realization. So in total, 700 animals were sampled in 79 farms in 74 villages distributed in 10 departments of the 3 regions that we did our studies. So this the chi-square test applied to the data show a significant difference between the prevalence that we are able to obtain by microscopy and the prevalence obtained using microscopy were 5.6% in the total samples, and the prevalence obtained by PCR was 12.3% also. For the abundance and geographical distribution, so we suspected four species of trypanosoma that we can say trypanosoma vivax, Trypanosoma congolens, Trypanosoma theileri, and Trypanosoma similae. But we have been able to identify 3 species of trypanosome. These were Trypanosoma vivax, Trypanosoma congolens, and Trypanosoma similae. So, part of these 3 species, we can say that the most abundant is the Trypanosoma vivax, followed by Trypanosoma similae, and finally by Trypanosoma congolens. So, according to the distribution of the Trypanosoma vivax, as we found that in all of the departments of in our sampling areas, and also we have found only in 2 departments, only in Trypanosoma vivax, Trypanosoma congolens were found only in 2 departments, as in Bengue and Kutu. So in this study, we are not able to find the Trypanosoma brucei, which is also commonly found in hairy cross. As this high proportion of Trypanosoma vivax is a concept with some observation made by several researchers like Akapovi, Boka, Dayo, and those. So the dominance of this Trypanosoma vivax could be explained by the presence of some sessile like Tachinoides, Palpalis, Gambiensis, and some BT fly also. So here we have a comparison between the breeding, the different breeding. So we can see that the Ndama breed have the most important prevalence, followed by Mary, which is a crossbreeding between Ndama and Zebu, and after we have a Zebu race and also Bauli race. So, as a dose factor, as a region, age, and sex, we can say that except race, all of this factor were statistically different between the modality. The infestation of the young animal under 2 years of age was low compared to those to over age of group. So, here the young animals are more closely for monitoring for the health problem. In addition, they are kept on the PK all of the day in the park. So they are not, they are less in contact with some beating fly and also some vectors that they could transmit in a HHA fly, trypanosome. So here, using the model of microscopic diagnosing infection, only Park Severi had a significant relationship with the infection. So here we can see that we have a relationship between infection diagnosed by microscopy and the Park Severi. So we can see that the anemia of the percentage of Park Severi is an indicator of the health status of the animal risk for the fieldwork also.
You need to conclude.
But when the Park Severi has low percentage, the prevalence is high. This result confirms our position on that we have a close relationship between Park Severi and whatever not the animal is infected. So depending on the species of trypanosome responsible for the infection, we have a significant observation. The significant difference was observed between the myth of Park Severi of the animal, which is infected by Trypanosoma congolens, compared to. So here it is we can see the pathogenicity of Trypanosoma congolens compared to those Trypanosoma. In conclusion, we can say that our finding is indicate that bovine trypanosomes is prevalent in Côte d'Ivoire. This prevalence has a variation to one region to another region of this pathogen, include Trypanosoma vivax, Trypanosoma cineathambo, and Trypanosoma convoluens. So, contrary to those species, Trypanosoma convoluens were only observed in 2 departments as Mbenge and Puto. And PCA still the more sensitive technique for trypanosoma detection. And the parcel volume also remains a key indicator in the process of rapid diagnosis of trypanosome. So, for the end of this presentation, we want to acknowledge World Bank, PASEC, ECP, and Sokoine University, SACI, WPI, Pelefo University, Institut Pierre Richer, and Lanada. So, thank you for your keen attention.
Thank you very much for that presentation. It's good to see RSIF represented. Let me call upon Samuel J. to give a presentation on the Continental Atlas in Nigeria. Samuel? Okay, is D. Barte here? Please come and present. So we shall not have 3.13 because of an incident where she lost her laptop, which is quite unfortunate. Yeah. So please go ahead and give your presentation.
Bonjour à tous, je suis Bertrand Diakarija de l'Institut Pierre Richer de Bouaké en Côte d'Ivoire et j'ai le plaisir de vous présenter les résultats préliminaires sur l'élaboration de l'Atlas sur les CC et les trypanosomoses en Côte d'Ivoire. Ok, merci. Pour situer un peu le contexte, il faut juste rappeler que, comme dans bien de pays, la trypanosomose animale constitue l'une des contraintes majeures au développement de l'élevage en Côte d'Ivoire. Le pays en est endémique et c'est la zone nord du pays, représentée ici par les couleurs vertes, qui en est la plus impactée. Et cette zone abrite 70% du cheptel national. Nous disposons de données sur la présence des glossines et des trypanosomoses en Côte d'Ivoire.
Breaking.
Certaines ONG ou dans le cadre de certains projets. Malheureusement, nous n'avons pas.
Unfortunately, we don't have synoptic information on trypanosomiasis, and the absence of this panoramic vision limits the possibility of a rational planning of control, and it also makes it difficult to estimate the eventual impacts. So there's the need to develop an atlas on trypanosomiasis and also on tsetse flies in Côte d'Ivoire. So as to carry out this study, we followed the same methodology like other countries before us, which ended in the development of the atlas. There's Mali, Burkina Faso, and Kenya. We can summarize the different, we can there are 5 steps in the development of this atlas. The first one is creating of the national repository, which is then followed by the processing the data, and then the building the national geodatabases, and then there's the creation, the generation of maps, and finally the generation of the documentation. Concerning data collection, we started, but what we thought was the most available, which we have. We targeted the data which had been collected in the activities of Pierre Institute between 2005 and 2022. The intervals can vary according to the abundance of data collected. The data are not yet published, and then also data from scientific publications, and also mainly from entomological survey data, especially our structuring, the structure of our digital repository. We have these ones, and in each dossier we have the data for Côte d'Ivoire, and we have the different dates for the collection of samples. The the data processing, it was organized by zone and by data. It was there were corrections were made. There was verification, standardization, and then it was put in the repository, the data repository. After that, we created another database which we cleaned and harmonized. This map shows the zones covered by the data we collected. Out of the 31 regions of Côte d'Ivoire, the data covers 19 regions. We observe insufficient coverage of the northern zone, which is the main area for livestock in the country. With preliminary results, we have counted 3,000 trapping sites, which is more than 3,500 trapping events and about 9,000 trap days. More than 21,000 flies were captured belonging to 8 species. We have to note that these are the same species which were described about 40 years ago. It has not changed so much, the glossine. So the 3 groups were captured. There is Glossina mositons for the mositons group, which are localized essentially in the central part of the country. We also captured the Glossina of the Fusca group in the forest in the western part of the country, and also in the center of the country in the forest of Mariwaki, which
Sorry? Oh, okay. So we are shifting now to 5C on diagnosis. Yeah? Yeah. So please introduce yourself.
Bonjour, moi, on m'appelle Eustache Onyema.
I'm called Mustache. I'm a doctoral student in Benin, but in these activities we are in IPR in Cote d'Ivoire. Thank you. This project, which we are going to present to you, is the project combat project which we have heard about in this conference. This project is entitled Design and Production of 5 recombinant multivalent proteins for the serological diagnosis of trypanosomes, African animal trypanosomes. I don't want to come on the problems which represent trypanosomiasis in Africa. This is well known, but I would like to put emphasis on certain issues like problem of vaccine and also vector control, chemotherapy, trypanotolerant livestock. So it's necessary to develop effective, simple diagnostic methods, and which is not so expensive. Here, I'm showing the existing tests. I'd like to draw your attention to a serological test, especially test ELISA, and for detecting antibodies. The detection of antibodies are not synonymous necessarily with a confirmation. This test is used for monitoring control campaigns. For ELISA, for detecting antibodies, I can regroup that into 2 groups. The first group is that recommended by, which is based on the total protein of trypanosomiasis. This test is known for its relative sensitivity and also there's also ethical considerations. There are also problems of standardization. ELISA is not the same as another one. We also said that this project also would like to improve upon this test by the culture of trypanomyces. Then there's the same type of test, but this time it is based on recombining antigen, and it represents the most adapted test. Only that there's lack of sensitivity. Our hypothesis is based on the following facts. On one side, we have a test recommended, well known for its sensitivity, and on the other hand, we have a test based on recombining antigens. So it will be possible to have a compromise between these 2 tests by associating recombinant proteins, which can also find with possibility to increase its sensitivity. This type of adaptation can be done 2 ways. And the problem of standardization will always be there, and therefore we propose the schema format. A schema is a protein which is constituted of immunogenic determinants, which is used for serological diagnostic. Our objectives and also the method we have used. As I said, we hope to develop a test ELISA based on chimeric recombinant antigens. Now, to succeed, we have already done the literature review so as to identify the candidate, and these antigens were analyzed, and we selected and constructed seven chimeric proteins. We had to purify them and then afterwards validate the test by determining the different parameters of the test. What are our results? More than 2,000 articles were in 66 were analyzed, and the fact that there's no homology with the others, these antigens have been tested using ELISA, using target species, that made us to arrive at 24 proteins which can be analyzed using bioinformatic tools. And so all that now made it possible to select 11 candidates in the immunogenic regions. We constructed this diagram. There are about 5 of them, and it varies. The size varies, and it is between 6. The number of proteins used to vary between 3 and 5, and the number of regions between 3 and 11. I don't have the time to go into detail of the specific structures of this chimera. So this is some kind of success. This protein was expressed, as you see on my left. We have expressed them and we purified them by chromatography. Here we have tried to determine the optimal concentration of antigen and of serum. And here this purified protein, 4 purified proteins. We used a protocol, and we incubated 2 pools of serum, one negative pool and another positive pool, as you can see on these 2 figures. The serum recognizes our protein with some degree of sensitivity. Here we tested the 4 proteins on a panel of 16 positive sera and 16 negative sera. As you see, there were at least 2 proteins where the results are better, at least, and which distinguish well the negative sera from positive sera. And the 2 other bad results may be attributed to the problems of purifications of antigens. Our conclusions: in this project, we attained our objectives in terms of the production of 5 chimeric proteins from 11 proteins. They were tested, but we need to improve the quality of the purification of these proteins, and more experimental sera have to be tested in the coming days. we should continue testing these proteins, and where we shall improve upon their purification. And then we need to validate the test parameters by comparing them with the standard test which exists. We have also thought of there also the yeast system which we want to do. Thank you.
We now move on to Fabrice. Fabrice will talk to us about characterizing the evolution of basic variables. And you can tell us more about it.
Okay, thank you, moderator, for the speech. Before I begin, I'm Fabrice Somé from Burkina Faso. I'm pursuing a doctorate degree in human biology, and I'm very happy to share with you my preliminary result based on characterization of the evolution of basic hematological and biochemical variables in relation with bovine trypanosoma during an experimental infection by Trypanosoma congolensis. With your permission, I will shift in French where I'm more comfortable to make my presentation.
Okay. In introduction, we can say that in the areas of high pressure of Glossina, there's a bovine population which can be divided into two groups. We have here the first group is the group of animals which are sensitive to infection. These animals cannot survive without treatment, medical treatment. We have to realize that today, with the emergences of isolates, and then the second group here, these animals which are tolerant to trypanosomiasis, even if they suffer from the disease, they have the capacity to develop mechanism so as to limit pathological effects of the disease. A number of studies were carried out so as to identify the biological mechanisms which are associated to this tolerance. And we found out that this is a multigenic trait, and the different studies carried out so as to identify the genes did not really allow to have results which are conclusive, especially the relationship between biochemistry, immunology, hematology, and tolerance. In cooperation with CIRAD, we did a molecular study, and we saw some different and from these recommendations, it was decided to do another study so as to be able to identify the biochemical and hematological variables which is related to tolerance with trypanosomiasis. We did another study, and this study was done in Burkina Faso. We used 3 species. There's the first one, which is Damas, which is resistant. The 2 others are the zebu and also crossbreeds. During this experiment, we infected all these animals with the isolate congolensis, and the infection was done. For hematologic parameters, here we had leukocytes, red blood cells, plaquette cell volume, lymphocytes, granulocytes, and we used For biochemics parameters, we measured cholesterol, and we also used a tool called CabioSync. During the whole study, we also studied the dynamic of functions, parasitomology, and the study took 6 months. As preliminary results, you can say that the dynamic of this parasitology, and where we have a rate, we observed, we saw that the infection dynamic through parasitology in all animals was very high during 3 months. It's after 3 months that there was a fluctuation in reduction and increase, and this lasted for the whole period of the study. We also have to say that what's important is that during primo-infection, the tolerant animals were not able to control parasitology. What is interesting to note here is that these animals have the capacity to control anemia, and what is put in green there, they were able to be tolerant, whereas others presented severe anemia. So we also have to note that 1 month after infection, one of the animals died. Here we do visualize PCR, physical component analysis, the different variables. As you can see here, parasitemia is negatively correlated. That is leukocytes, lymphocytes, hematocrit, and red blood cells. Now, with this correlation, it's a negative correlation. That means that when these hematological and biochemical variables increases in the blood of the animals, there's a reduction in parasitomology. Then there's also a visualization of the relation between the different animals. As you can see here, animals which are tolerant to trypanosomiasis are regrouped on my right, which is in relationship with the variables. These animals, during infection, they produce more biochemical and variables, and the animals which are sensitive to trypanosomiasis, they have a level, a parasite level density which is weaker. In conclusion, we can say that this study made it possible for us to measure the major difference in terms of cells of and there was also a high reduction. This study makes it possible to suggest a complex dynamics between animals and the parasites. And lastly, here we can see that there's a strong relationship between immune response, and what we have to retain here is that during this study, we also explored the immunological parameters, as you say, the response. We collected serum, which we are going to dose very soon with antibodies, and then the stockings which are produced during these major actors. We also did a cellular analysis. We studied the cells through stometry so as to see the cells which were not being detected so as to be able to identify clearly the immunological factors linked to the disease. I'm going to conclude my presentation by thanking the European Union for the support within the framework of this project, Compact, and also I thank CIDAS, which made it possible for me to carry out these studies. I also thank CIRAD, which also gave me a lot of opportunity, and thanks to that I was able to present to you these results. Thank you.
Thank you very much for your presentation.
Merci beaucoup pour votre présentation. J'invite Ola Wale de la Gambie, s'il est là. Ola Wale de la Gambie. Il n'est pas là. Maintenant, c'est ça. Youssef du Soudan. Youssef du Soudan. Bien. On va nous parler de la détection Sherlock.
Hello? Hello?
Okay, we'll have a change there. Olawale, you can go ahead and give your presentation. You have 10 minutes.
Okay. Yeah, good morning, everyone. Can you hear me?
Yes, we can hear you.
Hello?
We can hear you.
Okay, thank you. Yes, this is Professor Solano from the Gambia, and I'll be presenting the title of our work, Molecular Microscopy Diagnosis of the Gambia Cattle for 3 trypanosomes. This work was done under the supervision of Professor Dr. Ibrahim Kaya, who is also joining us online from Turkey this morning. Yeah, these are the contents of what I'll be speaking with you this morning about. So I'll be talking about the introduction to our work, the objectives, the materials, and the findings, and I will round up with a few conclusions from the work. So our objectives are in 3 folds. We focus on the naturally based cattle in the Gambia, and first we wanted to know what is the existing prevalence and the distribution of 3 trypanosome species, the T. cruzi, T. congolense, and T. fibrans, based on 2 methods, microscopy and a more recent advanced method based on polymerase chain reactions. Also, we want to provide solutions concerning the challenges that smallholder farmers face in control and eradication of this African animal trypanosomiasis. Eventually, through all these steps, we want to provide evidences to show that disease is still a problem among the smallholder farmers, especially in the Gambia and by extension in the whole of sub-Saharan Africa. So our study context is, this study was carried out in the Gambia, and this latitude and longitude as shown here. Gambia is in West Africa, as you may know, and one of the smallest countries in the mainland Africa. So agriculture constitutes up to 23.7% of the GDP, gross domestic product, and the climatic condition is Sudanese Guinea, usually with a short rainy season for about 4 or 5 months at most. And Gambia is unique because most of the animals, especially cattle now, are local breeds. The Dama constitutes about 97% of the total livestock population in the country. There are other ones which include the crosses between the exotic ones. But when it comes to the local ones, most farmers keep this in Dama because several studies have proven that this in Dama is tolerant to trypanosomiasis. So we did a study in 2021 based on meta-analysis, wherein we combined all the studies conducted on cattle, sheep, and goats. And specifically for cattle, we realized that based on this meta-analysis, there is an existing, the national prevalence as at that time was 5.2%. And we were able to also establish that there's a declining prevalence of trypanosomosis in the country based on that systematic review and meta-analysis. Just for emphasis, this is the Ndama breed of cattle, which is found in about 20 countries in both West and Central Africa. And as I said before, studies shows that a lot of studies have proven that this breed is tolerant to the disease that we're talking about this morning. So straight from our studies, we collected blood samples, but we didn't just start with that. We did extensive literature review, which part of it I've said before. Also, we did consultation with those people who are on the ground relating with farmers, which we call the livestock extension personnel. We also consulted with the scientists who have been working in these areas and also some other stakeholders to arrive at the study objective and how, when, and specific place to collect the blood samples. So eventually we arrived at 5 regions out of 6. Gambia has 6 regions, but we chose 5 of them where the disease have been proven to be prevalent. So out of these 5 regions, 4 of them we chose 2 districts, and then one of them we chose 1 district. That's why we have 9 districts. So in every district we chose 2 herds, 2 different herds, which we have 18 cattle herds altogether. So sampling were done in 2 different seasons. First in the rainy season and also in the early dry season, using those 5 regions and then 18 cattle herds in each of the districts. So while sampling, several other parameters were collected, several risk factors, the breed, sex, and so on and so forth. So altogether, in those 2 seasons, we were able to collect 921 cattle blood samples from 21 villages in 9 districts. This graph shows the distribution of how we collected those things. We can see there are 21 villages, and in some places we collected up to 50 samples, and the list collected from many of the villages based on the number of animals present is 11. So we had a condition for selecting a herd. At least there must be 25 animals in a herd before any herd is selected, and then we did random sampling when in a particular herd. So this is the way the blood were collected from the jugular vein of the cattle. And so the laboratory analysis, after collecting the blood, they were put in the cold pack and transported to the lab immediately. So in the lab, we did pack cell follow, which is a democratic determination. Also, we did microscopy using Murray et al.'s method. And then also, we did molecular diagnosis. For emphasis, we obtained positive samples from University of Glasgow in the United Kingdom. And we did manual extraction using Indiscrete quantitative kit. And we quantified the DNA by nanodropping, and there was a thermal gradient to optimize the PCR conditions. So all these analyses were done in the lab. And then so we did real-time PCR, and also we followed that with species-specific professional PCR. So altogether, there was 25 microliter total volume for each of the PCR that we did. And then, apart from the real-time PCR, we also carried out capillary and gel electrophoresis. So, the results that we obtained out of it. So for the PCD, the plant cell volume of the sample animals, we categorized them based on less than 20, 21 to 25, 26 to 30, and the 31. So this was based on the literature on this aspect. And in the Gambia, there's an international tolerance center, which I believe most of the participants are familiar with, though it's defunct now, has changed to West African Livestock Innovation Center. So this is where part of this study was carried out. And the study done in this center before shows that any animal that has PCV less than 20 should be treated for trypanosomiasis, even though they are asymptomatic. That means they don't show signs, they should be treated. So our study confirmed is that most animals that are positive for all the species that we looked at have PCV less than or equal to 20. And as we can see from this table, we couldn't see any species by microscopy. This result is based on microscopy for T procyte, but for T congruency and T fibers, there were some animals positive for that. And so based on the body condition score of those animals and then the season of sampling, we used 2 seasons, like I said before. So we have the rainy and the early dry. And then we see that the highest prevalence was in the rainy season among the lean animals, using these 3 categories, fat and medium. And then when it comes to the lowest prevalence was also in the rainy season, but that was among the fat animals. So this is the graph showing the amplification of our positive control. So we had a good amplification, and the positive control control has the toll-like receptor 8, which we used just to show that we got right. So this was for the T side, the amplification curves that we had, though it was late, and but we were able to get some amplifications for that. So we analyzed the whole sample based on that, and we got positive control for our T side, but for the pitch samples after DNA extraction and the PCR, we couldn't get true amplification for any of them when it comes to T side. So the same thing, and then we also try to analyze that with a conventional PCR gel electrophoresis. So similar results were obtained, positive control amplified, but for the big samples, no amplification. So the same thing with other samples that we tested, this for the T fibers and T covalency, using 2 base pairs, 100 base pair ladders and 50 base pair ladders, just to make sure that we did a lot of things to rule out any doubt. So eventually we got 0 prevalence for that, and there was no amplification. So in the final results, because we didn't get any positive sample through microscopy and also through real-time PCR and conventional PCR, so we concluded that in the Gambia, we could say to a very good extent that there is no T procyte in this case, which is present in cattle. Okay, for the among the 921 samples that we collected, we could say that we couldn't find any T procyte by all the 2 methods, both microscopy and molecular diagnosis. And I'm thankful, one of our presenters this morning also emphasized that in one of the countries that they couldn't see any T procyte. So for the T fibers and T covalency, we got positive samples by microscopy, but not with the real-time PCR and the conventional PCR. So this was something of interest to us, and which called for a lot of investigation and a lot of consultations with people, because by microscopy we have positive samples, but by other more sensitive methods, we couldn't get positive samples. So we suspected a lot of things might have done. Possibly there were cautions that we need to do or some things like that. So with conventional PCR, we said none of it. So conclusively, we said for T fibers and T components, our results were not conclusive on that because there was no congruence between the microscopy at the same time the other more sensitive method, which is the PCR. So what are the implications of our findings from this study? So we suspected the DNA kit used possibly was not very sensitive for the extraction. So we contacted the company, but they proved to us there's some other people using the kits and they were getting positive results from that. So looking inward, we come to this conclusion from the study that we need to, or for other studies also, there's a need to pay careful attention to these parameters, especially when it comes to the PCR, the choice of the probes and primers, the thermocyclers used, and then the denaturation and the optimal condition for PCR. There's a good need to pay attention to all of this. And then we see that there's a need to consider diversity among some trypanosomes. So in the Gambia, we suspect that the T fibers and the T convalency that we have in the Gambia might be different from what we are seeing, because most of our animals are trypano tolerant. So which calls for sequencing. We need to sequence those positive samples that we got by microscopy, but there's no present funding to do that. So it's part of our future plan, and then we'll be able to come up with the exact primers and probes, so that we can be more efficient with the PCR in the future. And so to that, they have said that, so we need to do the genome sequencing. So in conclusion, we could say that based on microscopy, we have some positive samples of T convalency and T fibers, but not for T cloci, based on three methods: the Bobby-Cole technique, the real-time PCR, and computational PCR. So we could purportedly establish there's no prevalence or no prevalence of T cloci in the Gambia. So for T convalency and T fibers, our results were not conclusive because we couldn't find congruence between the three methods used. So we want future studies to improve on the molecular diagnosis of T convalency and T virus, and we're open to collaborations in this regards. And so we also want the geographical atlas of cystic-like trypanosomosis to be done in the Gambia, or to be performed, because the last information at the national level from the Gambia was done in the 1980s. And that's what we still have up to now that we're still working with, where the country was divided into three regions, into three aspects: high, medium, and low trypanosomosis challenge areas. But we know things have changed: demography, climate change, and a lot of factors have come in. So we really need to update that. Okay, and there's no, to the best of our understanding, there's no patent implant in the Gambia at the moment. So a region where trypano tolerance studies started from, with the International Trypano Tolerance Center established in this country. So I would bet urgently that Gambia should be put on the patent agenda, and this is an opportunity to ask for things to the people organizing this, to put us and consider us in the patent. Also, we highly solicited for partnership amongst several stakeholders who are participating here, or may later know about this. So several organizations were involved in this study, both in Turkey, in the Gambia, in the United Kingdom, and I do acknowledge payment to them. So thank you for your attention.
Time.
So I would like first to thank the organizing committee that allowed me to share with you this work today. So I am Roger, and I'm currently working with IRD and Pasteur Institute in Paris and Pasteur Institute in Guinea, and also with the great support of the Programme National de Lutte against the Neglected Tropical Disease in Guinea. So today, I will be speaking about how we developed and applied the Sherlock molecular detection in epidemiological surveys of animal African trypanosomiasis. So this work is included in the SHERPA project. So you can see the logo here. And in this project, we have developed a novel detection, so molecular detection toolbox for all of the trypanosomes of human and veterinary importance. So first, we have developed a broad assay that is able today to detect all the trypanosomatids that are circulating in the animal world, and we are even able to detect trypanosome insectiflies. And besides this broad assay, we also have developed species-specific assays that are able to distinguish between every species that are listed here. So, in practice, what is Sherlock? Sherlock is a CRISPR-Cas-based diagnostic. So CRISPR is the molecular scissors, so that are able to cut total nucleic acid, I will say. So this method is mainly divided in two parts. So you have the first part, which is the amplification. So the amplification starts with an extraction of of a total nucleic acid from blood, dry blood spot, or skin samples. And after this extraction, we will have the amplification of the target. So this first part of the Sherlock assay gives us the sensitivity of the method. And we have the second part, which is the detection part. So in this detection part, we will mainly have the specificity of the assay. So in this detection part, it's in this detection part that we will be using the molecular scissors that is able to cut a reporter. So when this reporter is cut, you have the emission of the fluorescence. So the fluorescence is just meaning that you have the target that you wanted to detect in your sample, so you have the trypanosome that you wanted to detect in the samples. So this fluorescence it can be read in the lab, so using a plate reader. So this plate reader, with this plate reader, we have a high throughput assay. So because in each plate, we are able to analyze 386 samples within one plate. So we are using this in the lab, but our plan is to develop a lateral flow assay that is more field adaptable. So one thing that is really important here is that this assay can be run isothermally. As you can see, all of the steps can be run at 32 degrees or even at 37 degrees. So there is no need of heavy lab material to run this technique. So for trypanosomatid species, we have developed a lot of assays. So the first one is for all of the trypanosomes that are circulating in the animal world. I mean, all of the trypanosomes. So this assay also includes Leishmania, for example. So you can see that the limit of detection of this one is 10 parasites per ml. And you also have an assay that is able to detect all of the trypanosomes with the same limit of detection. And you can see that we are developing some assays so for specific targeting the parasites. So you can see that species-specific assay have limit of detection that vary from 100 to 10 to the 4 parasites per ml. So if you want more detail about the techniques, so don't hesitate to come and speak with me or go and see these references where we explain a lot of things inside this article. So now we have developed all of these assays, so we took all of them and we want to analyze samples coming from epidemiological surveys in a domestic pig in forest in Guinea. So here I show you the map of Inzerikore. So Inzerikore is located in South Guinea. And just to remember you, Inzerikore is an historical focus of HAT. So the last reported case was in 2004. So this is really an historical focus of HAT. So we went and collected in the green zone, we collected a lot of samples. So in this zone, you have a lot of farm. So we collect 200 samples coming from farm pigs. So you can see in this picture how this farm looks like. So we are mainly focusing in dry blood spots. So we use these dry blood spots and we analyze them with our method. So here I show you the result. So the result of this analysis. First, I show you the result with the pan-trypanosomatid assay. Here in the y-axis you have the fluorescence, basically, and the dotted line represents the threshold. So over this dotted line you have positive samples, and under this dotted line you have negative samples. I just want to remember you that this pan-trypanosomatid assay is to detect all of the trypanosomatid. And when we want to go further in the characterization of these samples, we see that we have 45% of positive for trypanosome. And you can see the pattern. You have a lot of samples that are highly positive, and we have samples that are positive but in the middle. When we wanted to detect, to look for TVVAX in the samples, we were not able to find any positive for TVVAX. And this was also very encouraging because we know from the literature that TVVAX, so pigs are not susceptible to infection with Trypanosoma vivax. So we cannot find any positive in their samples. So for T. Congo, we are analyzing, so when we were analyzing samples, we noticed that we have 9% of T. Congo positives in the samples, and almost all the samples here are co-infected with trypanosomes. And last but not the least, we were looking for also TB Gabianse, and we observe that we have 1 positive in the samples. So we want to go and to analyze the samples further to see what happened in this location. So here you can see the result of the mini-colon, the mini-ion exchange centrifugation technique. So you can see that the parasites are dancing in these animals. So when we compare the result of our Sherlock assay and the parasitologic data, so out of the 200 samples, you have 41 samples that were positive to the cat. So on these 41 samples positive to the cat, 19 of them were positive to the parasitologic data with this kind of parasite inside. So you can notice here that Sherlock is not missing any positive samples. So here I just compare the result of Sherlock pan-trypanosomatid with the results of the parasitology. We can notice that when we want to be more focused in the trypanosome and T-Congo, we have 3 samples that are not positive for both. And their samples are not also positive for TVVAX because we see here, we don't have any positive for TVVAX. Donc, their samples highlight the fact that we need to develop more assays to be able to detect some parasites that we know that they are circulating in these animals. So parasites such as Teleri, Simiae, or Suis, or even other parasites. So at the end of this presentation, I just want you to remember that with Sherlock, we have a highly sensitive and specific assay that is today able to detect active infection because we can focus on detection with RNA. And with this assay, we are able today to be accurate in the determination of trypanosome species in an infection. And we have now a promising epidemiological tool that will be helpful to monitor the reservoir host and the emulation status. So I was here alone to share with you this data, but this is a collaborative study. So I want to thank all of the partners that have been involved with this project, and I want to thank you for your kind attention. Thank you.
Thank you very much for your presentation, Roger. All the presentations from this morning are now open for discussion. So if you have a question, please raise your hand and indicate who the question is for. There's one there. Is there another? 2, 3. Let's start with this 3. You're number 3.
Oh, bonjour, Dr. Muyolo.
Good morning, Dr. Muyolo, RDC. My question is to Fabrice. If I have followed very well, you concluded that a group of animals and the other one were not tolerant. So my question is this. Is it an indicator for trypanotolerance and another one not having it? So the question is to Fabrice. Alors, Fabrice, vous pouvez...
Then we'll go to Merit.
Yeah, thank you very much. My question goes to the, I didn't catch his name, to the Burkina Faso person who compared the different livestock breed. So he showed that this indema, even if the parasite still exists, but managed to control the PCV. So the question I have, of course, has a lot of things to do in the future, but do you think the resistance is at the red blood cell or not? That is my question, because you know, if you see the trips and red blood cells interaction at live, you can see the way they attack it. It's so that how they manage to control the anemic level despite they have high parasitemia. Thank you.
Okay, that's a question to Fabrice as well. So let's have the third question.
Merci beaucoup pour la parole.
Thank you for giving me the floor. My question is to Ivory Coast. I'm sorry because I don't remember the name of the presenter, but the question is to Ivory Coast. He was talking about the classification of the glossine. He was saying that this classification was either glossine purpuralis. It will depend if you are in the north or in the south. And he concluded, if I'm right, that now in Ivory Coast there is also a work which is ongoing, so they can also classify these species and subspecies. I want to know the kind of work which is now ongoing. It is especially focalized on according to the area, or is it a work which is based on the taxonomy, following the characteristic of the species? So, is it? Do we have a classification which is geographical?
John Kabaya will come to you after that. So Fabrice, and then the last question I think the last question was to Bate on the Atlas. Yeah, so let's start with, where's Fabrice? Okay, let's start with the last question.
Ok, merci beaucoup pour la question.
For the question, what we have to say, this limitation was done 40 years back. It was based on the difference between the male, and the studies was done 40 years back. So north and south. So the parameters were also respected. So the limit was defined. So the one which was in the south, it was Palpalis. In north, it was Palpalis gambiense. This limitation can change according to the variation from the environment. What we are doing now, when we are doing now these studies, on top of these observation of parameters, we have also the PCA. They have also to amplify the genes. In this way, you can now do some kind of classification. And now about the female. There is also a difference between female and male. So these are the different elements that we have also to consider, and can also able us to actualize now the result.
For the response, is Fabrice? Yeah. Okay.
Ok, la première question.
The first question which were now addressed to me, if there is an indicator about this disease, I don't know if I have understood it very well. We have also to say, through the different studies that we had now, we could not identify a difference before the infection to say that this animal it could be sensitive. So this tolerance, these animals, you have also to infect them. You have to observe them during a certain period of time. So it is very difficult today because we need also some studies. If we can identify this character biological, so before the infection, so we can say this animal is trip tolerant. When you're trying now to verify these also factors.
Are able to control anemia while harboring high density of parasitemia. I don't know if it is okay. For this question, I can say that to control, there is no link between controlling controlling anemia and parasitemia. And this aspect, controlling anemia is like a natural phenomenon. And to control anemia to control parasitemia in the first time, it's really difficult for the animal. The animal need to be reinfected, and then the combination of immunology response and also another factor, maybe biochemical or hematology factor that can allow the animal to control the parasitemia. So to conclude, I will say that there is no real link between control anemia and parasitemia.
Okay, thank you very much. I think there are some questions online. Please check. So there's a question to Olawale. Sorry, I'm not. Olawale, there's a question to Olawale, there's a question.
Yes, I'm seeing it. The person was asking whether the DNA extracted from the blood sample was positive for T virus under microscopy. Yes. Based on the Murray et al. 1983 that I cited, which is based on the morphological appearance of these strips. So we said they were positive on that, and this was confirmed by people who have been using microscopy for a very reasonable length of time to show that this was for T virus and this was for T congruency, and that's for the microscopy. So for the PCR, we, like I said, because we got our reference in the primers from a publication by I think Ahmed 2018, which was a study conducted in Burkina Faso. That was where we got the primers from, and they sent it to a company to do it for us, and which they sent back. So very sure whether those primers that were used for T-Vivax and T-congruency in Burkina Faso, because there could have been changes in the structure of these ones that we have in the Gambia, and that's why I said, because we have a unique population of cattle in the Gambia, which is the Ndama, the Chapano tolerant, constitutes about 90-70% of the national livestock, according to the National Livestock Population Census in 2016. So that could have been, we are suspecting that there's a little bit of difference when it comes to the DNA of the tribes that we have, the trypanosomes, T virus and T congruency, that we have in the Gambia. And that's why we really want to do the genomic sequencing of those ones that we identified by microscopy. I hope I'm able to answer the question.
Okay, thank you very much for that response. I think we'll go back to a round of questions, starting with John Caballo.
Thank you very much, Mr. Moderator. And my question goes to the last presenter, I think it was Roger Jr., on using Sherlock in the epidemiological surveys of animal trypanosomiasis. I wanted to, he mentioned that there is a limit of detection of 10 parasites per milliliter. And I wondered whether for such a specific and highly sensitive method, what that means. Is that 10 different parasites, or is it a question of the the concentration of those parasites, or is it, because it would point to the possibility of interference, or can you elaborate on that and why there is a limit of detection, and what does that mean? Thank you.
Okay, we have a second question.
Ok, ma question c'est à Nancy Kentek.
Is now for Nancy, my question. For what you have developed, I want to know in the area where you have tsetse flyers. We don't have also control of flyers. We have also the veterinary service. How do you do now the classification? It is also a tool of controlling the disease. So you don't control flyers of tsetse, and we have also some treatment you are giving animals. Alors, David.
Yes. My question is to the last presenter on Sherlock. Just a few clarifications. What samples are were they using and handling? How are they being handled? And then is there a kit that is available? And how widespread is this test actually outside of your environment where you did the test? And then the other thing is I noticed in your fluorescent, there's a lot of moving parasites. How do you distinguish between the different the trypanosomes amongst that kind of sea of trypanosomes?
Okay, thank you, David. Johnson Oma, you have a question?
Yeah, my comment was to Olawale, I believe. That is the guy who presented from the Gambia. I think he's right to hypothesize that there could have been changes in the surface proteins of trypanosomes, and that might have affected the detection by PCR. So this actually, John Caballo alluded to this yesterday when we were talking about, sometimes maybe the difficulty in detection. I think the suggestion to sequence would be an ideal one, and maybe, design primers now that are specific and do a comparative study to see. I think it will add to the knowledge base of what we are dealing with. What happens when trypanosomes, you're dealing with trypanotolerant species versus susceptible species, and what that does to diagnosis of the parasite.
Okay, that's a comment. Thank you, Johnson. Roger, are you ready to respond to those two questions?
So, thank you. Thank you, Professor Caballo, for the question. So about the limit of detection. So this limit of detection that I show in the slides have been established in the lab. So we are using a lot of parasites, and so we are diluting them until we are not seeing them anymore. So, and after we took the samples and we analyzed them. So we are able to detect the dilution until 10 parasites per ml. So that is not meaning that, I don't know what happened in the field. I don't know if we are able in the field to detect until 10 parasites per ml, but in the lab we are able. And we can even detect less, but it's not sustainable, it's not replicated, it's not repeatable. So here, what we know that we are able to detect until 10 parasites per ml in the lab. And about both other questions, so the samples that we were using were dry blood spot samples. So it was just drop of blood in the Watman paper. So to handle this is just handle at room temperature with this silicate bead, so to let the samples really dry. So when you have this sample dry, you can conserve them maybe for 1, 2, or 3 years, and we can analyze them after. So this is not a problem. And about if we have an available kit for Sherlock. So we are developing it, so we have all the solution. We are in contact with some industry, and we want to make a kit, but we are now in developing the method. So there's not available kit for the moment, but we can analyze samples. So if you are interested, we can discuss and make some.
Okay, thank you very much. Nancy, you want to respond?
Thank you for that question. I did not get the last part of the question very well. Maybe you could repeat. But for the first part on the areas where there is no SESE. Absence of SESE does not mean absence of AAT. And as such, we are classifying those areas as under stage 1, where we don't know the status yet. I did not get the other part where you're talking about the treatments.
Can I suggest that you catch up over the break? Because,
okay.
I think we'll bring this to a close, and I'd just like to thank the presenters. Please join me in appreciating the presenters for this morning. Let me now hand over to the chair, if he has some comments.
Thank you very much, moderator, rapporteur. Let us give a round of applause to the moderator and the rapporteur for the session. We are doing very well on time, and I do not want to spoil that. I only have a very short request that the rapporteur general, the general moderator, and all the rapporteurs, if you could please remain behind for 2 minutes. The Secretariat has something to discuss with you as you go for the coffee break. We have had a very, very stimulating session, incredible presentations. There were some, as I listened to them, I felt that when I grow younger, I want to be like them. So it just shows the quality of the presentations that we had. So I don't want to spoil that. Continue with the discussions over the coffee break. And again, let us be as obedient as we have been. Let us be back here at 11 so that we can continue and see that we maintain time. As you know, this evening we have been invited for a cocktail, and we want to break early so that we can also go and put our feet in some salty water. So enjoy your coffee break and see you at